Hi, it's Paul. Let's look at small cell carcinoma in different specimen types in respiratory specimens. First of all, sputum is quite distinctive. So the cells tend to be very degenerate. There's a lot of mucus in the background here, and you can see some very dark, small nuclei don't seem to really have much cytoplasm in them at all. There's a whole lot of debris in the background too, that grey material, which is necrosis. And then you can see these little dark conglomerations of cells. And even at this power, I think you can see that they're forming little chains. So they're more cohesive than lymphocytes would be. Here, see how they are wrapped around each other as well in places. You can see there's a moulding joint where the two cells meet there, a thin line. This is a little more difficult. In here they're more dispersed and so looking more like lymphocytes. You have to look for nuclear atypia and variation, and I think it's there, it's subtle. And make sure there's no lymphoid cytoplasm or other features there. See how even at this power, but they're forming like chains of cells in single file. Often they form these little aggregates of cells that are dead really, with necrotic material. But see out here in the open, we've got a nice vertebral column of nuclei moulded around each other in a linear chain here. That's very typical of small cell. And here a little vertebral column again, you can see that's called a ball in mitt sometimes, with one cell wrapping around one. Notice out to the right here, we've got a chain of better preserved cells. You can possibly even see that they've got stippled chromatin there. The rest are just pretty much pycnotic and dead, but very hyperchromatic. Now, bronch washes are usually better preserved than sputum, but not always. So the one that I've actually pulled out randomly actually isn't. There's a lot of macrophages here. We'll see there are some bronchial epithelial cells, but there's also lots of these groups of really hyperchromatic nuclei. And you can see the moulding in them again, scattered in amongst some ciliated bronchs and some pulmonary macrophages. Notice how much more hyperchromatic these nuclei are. But a lot of this specimen is a bit blown out. So here I can find a nice aggregate of them. And there is subtle variation in size and shape, they're just very small. And if we look at the morphology of small cell in FNA, generally speaking, the cells are much better preserved and it's much easier to see malignant features and neuroendocrine features. With an FNA, we should be just directly sampling the lesion, and so that's pretty much what you get. This is the air-dried Giemsa stain. We've got red cells in the background and lots of aggregates, really a monopopulation of one cell type. You can see that they're cohesive, they're holding together, and they're all quite hyperchromatic. In some areas, like I'm pointing out here, there's little rosette formations as well. And I probably haven't talked about that, but that is a feature of neuroendocrine carcinomas sometimes. On high power, the cells do have a bit of cytoplasm, but a lot of them don't appear to have much. And you can see nuclei, again, are moulding around each other. So they're forming little columns. If you see one, see that moulding area there between those two nuclei. There's a chain here. There's a chain down there of nuclei all wrapped around each other. And in this area you can see there's actually quite a bit of anisokaryosis, so variation size and shape, more chains of piled up cells like vertebral columns. And here is one of those Rossetti formations, so it's a bit like a glandular formation, and you can see nuclei wrapped around the outside, but they're actually moulding as well. These are the pap stain slides. Notice all the crush artifact here. These are very fragile nuclei, and that's often a feature of small cell. That's seen in histosections as well. And again, you can see it's all one cell population. And what we want to look for here, do you think, to make sure it's neuroendocrine? So on the PAPs, it's always handy to look for that stippled chromatin, which I can see nicely here, and make sure there's no prominent nucleoli present. Notice the moulding here, and again we've almost got a glove and mitt thing going on there, and the nuclei really wrapping around each other, quite small. So in summary, the major mimic of small cell carcinoma is lymphoma, and it can be problematic in some places, so it's important to exclude it. Sometimes immunohistochemistry and particularly flow cytometry can be handy in those cases. To rule out lymphoma, you want to look for that cohesion, especially those sort of chains of cells that are moulding around each other. You sometimes see moulding in lymphomas, but you shouldn't see terribly much. And check out the stipple chromatin inconspicuous nucleoli on the PAP. The air drives are always best to see moulding really, but you can often see it on the PAP as well. And that is small cell carcinoma in respiratory specimens.